study. I'll tell you a little bit about what I've been doing this spring. Why did I want to do an independent study? I got introduced to topics about water quality and environmental science in middle school. I did Science Olympiad. And then over COVID, I had this aquarium keeping hobby. So I kind of learned a little bit about nutrient cycling and what it takes to keep some plants and fish alive. And I wanted to kind of learn about how nature does that naturally in the environment. So my curiosity kind of drove me to do that. And then I was also interested in pursuing some sort of environmental science career. So I wanted to get some experience in the lab, kind of see what it was like. And then lastly, I got involved with a lot of my community groups. So I'm on the board of the Friends of Little Crum Creek Park, and they're a nonprofit group that maintains and protects the largest park in Swarthmore Borough, Little Crum Creek Park. So what did I do? I did a two-part kind of independent study. I was an intern at Willistown Conservation Trust. I was on the Watershed protection team, and Lauren was my mentor there. Willistown Conservation Trust is a nonprofit group, and they have 6 different programs, including bird conservation, land management, stewardship, community outreach and education, and the community farm. The second part of my project was my independent project at Little Crum Creek Park, where I kind of assessed the health of Little Crum Creek. So Little Crum Creek is a small stream. It is a tributary to the Crum Creek, which means that it is a really small stream. It originates in Swarthmore. And we can see that the Crum Creek is this long part here, and the Little Crum Creek comes in and joins it right before it deposits into the Delaware River. So the Delaware River is a drinking water source for millions of people across 5 states. And so looking at a small creek like Little Crum Creek gives us an idea of what pollutants might be going into the Crum Creek and then affecting the drinking water in the Delaware. The timeline of my study, I started off with an introduction to the basics of environmental science at Willistown, and then I learned how to properly collect data out in the field, and then take this data and kind of process it and understand what it meant. So then I used all these skills I learned from my internship and applied it to my project at Little Crum Creek. So the first things I did was learn about water and the connection to landscape. So land use plays a huge role in water quality. If you have a lot of developed land, then you have a lot of impervious surfaces, which is land that water can't infiltrate and soak into the soil. So you have a lot of runoff off of pavements and sidewalks that are all impervious. So all of the pollutants that might be on the road, like road salt, or the rain running off, and while it's getting run off, there's a lot of sun and it heats up the water. That all goes directly into the creek. And so if you have an area like Swarthmore that's very heavily developed, there's a lot of residential areas and retail areas, then you will have a lot of these issues reflected in the water quality. The second thing I learned was about climate issues through the book that I read called All We Can Save. It was a collection of little essays which kind of gave me a look about like kind of the individual perspective of how climate change impacts someone. So climate change is such a huge issue that you don't really kind of relate to a lot of the issues in it. So the collection of stories kind of put a human perspective on science. Well, I learned how minority groups are disadvantaged through like climate change issues. So areas that are near highways or areas that people who are low income are stuck living in have a lot of those health issues that are related to climate change, like asthma from air pollution. And a lot of what climate research tackles is kind of fixing those issues so that we can all live in a better world. And then the last thing I learned was a lot of opportunities in a career for environmental fields. Lauren has been in the academic setting, so doing research for a university. So I learned a little bit about what it takes to be a scientist in that setting. And then working for a government agency, in the environmental agencies. And then lastly, I learned the most about what it's like being in a nonprofit group like Willistown and doing conservation research there. So I learned how to do macroinvertebrates and monthly sampling. So here's some images from that. This is one of the preserves that Willistown manages. They take care of thousands of acres of land and protect it from development, and they do research on 9 different sites. So why do we look at macroinvertebrates? These are good indicators in the stream. Macroinvertebrates are any aquatic organisms with an exoskeleton that you can see with your naked eye, which is macroinvertebrates. They're abundant. They are in every continent except for Antarctica. So there are a lot of different species and a lot of different types. And they're categorized in different pollution sensitivities and pollution tolerances. So the main 3 species that conservation scientists look for in water are mayflies, caddisflies, and then stoneflies. If you find a lot of these species or these organisms in the creek, then that tells you that the water quality are to the standards that can sustain life for these organisms. I was also lucky enough to participate in the monthly sampling. I went to 2, and they basically start off 7 a.m. in the day. You get waitered up, you get all your supplies ready, and you go and hit 9 different sites. You'll get to a site. This is Ashbridge Preserve, and you will collect samples in bottles and different cuboliters. And then we also measure discharge, which is this tool where you stick it in the creek and it has a fan, and it basically records how much water is flowing through that area of the stream. And then lastly, we take a visual assessment, so we look at what's growing on the stream banks, and we estimate how much erosion is on the banks. So this is kind of an example of one of the data sheets. This is actually my data sheet. And you can see that we take into account what the weather conditions are and what they have been in the last 24 hours. So when we're looking at the numbers such as like nitrate or conductivity, we can understand if these numbers fit into what we were seeing through the weather and the temperature, which all play a role into what we're seeing in the chemistry. So I also got to be a part of the land management task, where we went to one of the other preserves and we treated hemlocks for these invasive pests called woolly algae. And you can see that we're injecting the tree with these syringe-looking things, which was actually really interesting. But we do this so we can treat the tree and protect it against the invasive pest that kills them. And we use this syringe method because this hemlock grove is near a waterway, and the more popular method is a spray that goes into the soil, and that has a danger of leaching into the stream. So that's the syringe method. And then in the lab, I learned how to do 3 procedures. I did the total suspended solids, chloride, and turbidity testing. Total suspended solids is basically looking at how much sediment is in a certain volume of water, and this gives you an idea of the erosion problem. So if you can understand how much sediment is within that 1 liter water, then you can use the discharge data and then compare that and see how much sediment is moving through that entire stream. So total suspended solids, I'll walk you through the lab procedure for this. So you take your sample container, you shake it up because you don't want any of the sediment to settle, and then you measure it in this graduated cylinder, and then you turn on the vacuum filtration, which has a little fiberglass filter which catches all the sediment in the sample. In the prepping for this lab technique, you need to rinse all of the equipment you have so you don't have any contamination. So everything that you see on the screen has been triple rinsed with distilled water first, and then you take the sample water and you rinse out those containers so you don't have any distilled water left over, so that you know that when you're doing the actual 1 liter measurement, you're getting only the sample water. So this is something I learned as being a part of the lab, is just how precise you want to be to ensure your data is accurate. So I'll talk a little bit about my project at Little Crum Creek Park. I had 4 different testing sites where I tested for the connectivity, and then LCC1 downstream was my main site where I did everything else. So you can see the unnamed tributary starts over here, that was my site, and then the most upstream is LCC3 with LCC2 before the unnamed tributary comes in, and then my main site is when they both join. So I did my sampling across 3 months, so I had 3 different sets of data points, and I took the sample and I tested it in the same day. So you can see that I was using the bridge as my lab setup here. In the field, I tested for pH, temperature, and connectivity, chloride, dissolved oxygen, nitrogen, and phosphorus. So pH takes a look at the acidity of the water. The pH affects how able the nutrients are to be absorbed by the organisms, and also how toxic the pollutants are to those organisms. The temperature affects the connectivity and chloride, which are the amount of salts, and the temperature affects dissolved oxygen. And if you have elevated temperatures, that can mean that you have a lot of stormwater runoff that is flowing to the creek. Dissolved oxygen is really important because the organisms need to have enough oxygen in the water so they can breathe. And then nitrogen and phosphorus are nutrients, and if you see elevated levels of those, then it's likely that there's human impacts like fertilizer runoff. I also did a brief macroinvertebrate survey where I walked along the main part of the creek, and we looked under the rocks and kind of assessed what the species were. So we found blackfly larvae, midge larvae, snails and worms, and then some net-spinning caddisflies, which are somewhat sensitive to pollution, but all the rest I listed were very tolerant to pollution, which matches the results that I found. So chemically speaking, we have normal temperatures for the spring right here, but then we have 15.3 and then the 18.6 as the outliers that you can kind of see on the chart, and the dissolved oxygen concentration kind of matches this. So you start off with some high dissolved oxygen, which is good, and then you drop to 6.58 here. And you can see through this chart that you have an inverse relationship between dissolved oxygen and water temperature. And then the rest, we have turbidity. Under 1 is what's recommended, so that's looking normal. Your chlorides here are a little bit elevated, and then the nutrients are stable, with the nitrate being a little bit high. So we can see the temperature kind of closely through this figure. The PA, code maximum, is this red line, so that's the maximum temperature that the stream should be, a side stream like Little Crum Creek. And these are the findings that I got. So all of my sites were elevated in temperature at every single sampling. So in March, this was the most significant difference, as you can see here. And then in April and May, there's a smaller difference, but they are all still above the maximum recommended temperatures. Another issue that I saw was that the specific connectivity was raised. So you can see the upstream sites, the red and the yellow, are the sites with the least connectivity. And then the unnamed tributary is super high up here. And if you could remember, the site where the unnamed tributary comes in and then joins, LCC1 is the most downstream. So you can see that as the unnamed tributary site trends upward, the LCC1 also goes up, which indicates that the unnamed tributary is affecting the water downstream. And when you look at the upstream ones, they're turning downward. So that makes sense because on May 8th, there was a brief rain, which dilutes the salt, so you should see lower connectivity. So what's happening at the unnamed tributary in LCC1 is that there's something washing in to the unnamed tributary, which then you can see reflected at the downstream site at LCC1. So after I did these results, I compared it to a historical data set that was at a site not so far from my main site here at Michigan Avenue. It was a USGS kind of federal monitoring station from 1971, and I found that most of the results reflect normal fluctuations, but the specific conductivity and chloride, which if you can remember, the salts in the creek, they have jumped significantly. So you start with a chloride concentration of 25 in 1971, and I found an average of 139 parts per million chloride. So there's a lot of salt in this creek. And this reflects that national trend where we're salting the roads a significantly amount more than we used to, even though the winter storms have actually lessened and we're getting less snow than usual. So what we can take from this data is understand that we don't need to salt the roads as much, and the board can use my data to kind of make recommendations to the community to make improvements to their own properties, which most of which lie along stream sides. So, so the board is able to take this data and kind of do future studies and look at where the source of this elevated salts are from. Some pitfalls to my study was it was only 3 months, so I only have 3 months of data, which means that a lot of the parameters I didn't really see trends in because I don't have enough data, but it did give us an idea that there are issues with the temperature and the conductivity that we need to kind of do more research on. And because my study was only in the 3 months in the spring, there's some seasonality to it, so it would be interesting to look at like dissolved oxygen in the summer when it gets really hot, and then in the winter it would be useful to look at the conductivity in the salts where there's more road salting and in preparation for winter storms. So overall, I learned that the scientific method is really messy. In doing research for this project and looking for historical data, there was a lot of difficulties in finding that data. I did contact like a Swarthmore professor, and we were kind of looking at if our data was comparable, but he did some stormwater collecting, which is different from mine, which I collected when the stream was in normal conditions. So it wasn't comparable, and another thing that I ran into was that the unnamed tributary and a lot of the upper reaches of my creek weren't even on maps. So it was hard to kind of find more information about those. But overall, with the internship, I was able to learn about all the different pieces about environmental studies, not just about watershed and watershed science. I got to understand how the land influences it and how kind of the environment is all these little factors that kind of come together, and they're all in this precarious balance that we're trying to preserve and improve. If you have a lot of salt, then it's toxic to a lot of the organisms, so fish can be like stunted in growth, and a lot of organisms who are sensitive, like mussels, will die because there's too much salt. Yeah, so it'll basically just make the stream less habitable. What is usually recommended is to put in like riparian buffers, which is just like plants that are near the stream that can soak up the water and kind of like make it so that the stormwater runoff or other runoff isn't going directly into the streams. Another thing is just more research, because it seems like there is a source in the unnamed tributary that is causing that elevated conductivity and those elevated salts. So the biggest thing that can indicate that the water is good is finding freshwater mussels. They're keystone species, which means that they kind of improve the environment that they're in. So they do filter feeding and they clear the stream of a lot of toxins. But because of that, when there's a lot of toxins in the creek, they're accumulating that and then they can die off. So finding mussels means that water quality is really great. So organisms like that and like otters, freshwater otters, finding those organisms are really essential because they're carnivores. So that means that everything that they're eating has to be present in abundance so that they can survive. As a part of this study, I kind of wrote a mini scientific paper to just summarize everything that I found. and give suggestions for what the board can do. So the Little Crum Creek Park board is going to be able to use this report and see these are the issues in the creek, and they're going to be able to also use it to apply for grant money to make improvements at the park. But also, a really important part of their job is having that community connection and being able to outreach to the people in Swarthmore and say, oh, you need to stop fertilizing your lawns right before a rainstorm, or you can get these native plants to plant in your backyard. And another thing that they're working on is naming that unnamed tributary so it can get on maps and be recognized. And to do that, because the stream is so small, it is technically private property, so the board has this really important job in reaching out to those homeowners and saying, you know, we're trying to do something good in this park, like trying to build that connection between the homeowner and the community